Okay, so Unit 4, down here at the bottom. So I have it all updated today. The only thing is I haven't put a link for this discussion board topic yet, but I will. So I just want to talk about this real quick for a minute. When you open up this file, it tells you about, we've been looking at some of the data, right? We've talked about incidents. We've looked at the number of cases of each of the infectious diseases, some of the ones that are reportable in the MMWR on the CDC's website. So this is a little introduction into that related to how they calculate these numbers. Even though it says here that you will calculate, I couldn't find my old file to be able to eliminate this. We're not doing, you guys aren't doing this. You aren't doing any calculations. You didn't do a case study this semester. So at one sentence, I can't eliminate because I can't find my original file. So, but I have a note there saying such. So we've been looking at lots of different graphs and diagrams, right? So this particular one is on listeriosis. And when you look at it, this should jump out at you, right? That there's a peak here. And when you look at the chart, it's in 2010, right? There was something that happened. Well, first of all, you have to know what listeria is, right? Which we haven't talked about it yet. But just in general, same thing for you guys, right? Your book is an excellent reference. So I referenced the book, so I just put the page number here. But notice that I went to the CDC's website and I did some further research to find out what happened, right, that would have attributed to that spike that you see right there. And so I give you guys an example, and this is true data, this is real stuff, real things that happened. It's not make-believe or anything, right? So that gives you an example of what I want you guys to do, right? I want you to look at the graph, and the graph that you guys are working on is this graph, right? So notice, again, right, we've got some peaking over here. Why is that, right? And it's pretty consistent each year, although 2010 seemed to be a little bit more, right? So what's going on here, right? Tell me a little bit about salmonella, right? And why we see these increases, and then do some specific research into these years. There are significant things that happened, right, that explain this trend that you see here, especially in 2010. Okay, if any of you guys can think back that far, you might actually know one of the things that happened. So, and then make sure that you cite where you got the information from. So notice in mine, I used 2 different sites, right? So I indicated and listed them. When you post in the discussion board your answer, then you'll be able to see other people's answers and be able to reply. You can, and it's the standard 2 replies, right? You're not going to be able to see anyone else's until you post, because your answers are going to be very similar, right? Otherwise, you'd be apt to maybe cheat if you could see everyone else's before you post. Does that make sense? So that's the only major difference with this particular discussion board, and I'll put a note to that. But you guys are working on salmonella, right? 2008, 2010, 9 and 10. What happened? Why do we see this increase in incidence, right? Number of cases per 100,000. And this list goes from like January to December, right? So January, February, March, April, May, June, July, August is up in here, right? Is what's going on. Something happened. Numerous things, actually. So read through this document and get started on that. I want you guys to try and have your initial posts done by Friday. So I'll remind you guys again Wednesday with the test so that people have time to reply before the following Monday. But this is a pretty easy one, right? It's not going to take you guys too much time. So we're on digestive, right? I also added the homeworks in here in this Unit 4 folder, although I'm thinking probably most of you guys won't do that until after the test on Unit 3. So finishing up digestive, last time I think we kind of ended in this progression of this topic of the mechanisms on how diarrheal diseases cause, yes, no, maybe so. Okay. All right. So if not, it's a good review. So number one is attachment, right? And so this is aided sometimes by the organisms having special appendages like papillae or fimbriae to attach. Others have other type of adhesion type molecules that allow them to attach. Some even will attach and secrete stuff to make attachment even more possible for even more cells, right? So this type of secretion system, and they cause even better attachment. Toxins are the big thing with these guys. A lot of them produce toxins. There's two main types of toxins. There are ones that cause us to increase secretion of water and electrolytes. So this is into the digestive system and therefore heading out of our body. Vibrio cholera is an example of one that produces those types of toxins. And then Shigella, dysentery, this one is producing toxins that actually kill our cells, right? They halt protein synthesis and kill our cells. And that's why with dysentery, remember the difference between gastroenteritis and dysentery. What's the difference with dysentery? What do you see in the stool usually, in the diarrhea? Blood, right? Because you're having destruction of the cells themselves, right? And it's usually due to the toxins produced by the organisms. Some of them use type 3 secretion systems, so they alter our cells. And this, again, this one really helps in the first one in aiding of attachment. But so once they attach, E. coli can produce these substances that it injects, like with the hypodermic needle, this protein appendage it has, it sticks it right into our cells. And it deforms our cells. Our cells in the intestines have microvilli, little tiny finger-like projections to the cells to help with digestion and absorption. What they do is the cytoskeleton inside our cells, the actin filaments, they cause them to rearrange in a way that it almost creates a platform for the bacteria to attach to, or a little pedestal. Some of them will also invade our cells, right? And again, a lot of times they're secreting substances into our cells and actually causes them to engulf. Not all of our cells do phagocytosis, right? Just our phagocytic cells, right? Our neutrophils and our macrophages, our dendritic cells. But all of them have that ability, they just don't use it, right? They have a cytoskeleton that they could manipulate if they wanted to. But that's not their main job. And in this case, they're activating that system within the cells and causing them to engulf them. And so Shigella is an example of an organism that does this to gain entry into our cells and actually grow inside our cells, which eventually, of course, leads to the destruction of the cells. So cholera, right? This one is caused by Vibrio cholera, of course. It produces a heat liable. This means that you can actually destroy this with heating, right? When you cook your foods, if the toxin is already present in the food, it wouldn't make you sick if you heated it. It can cause excessive secretion of water and electrolytes by the intestinal epithelium, right? This was the example that we gave. And what really gets people is that if you get too dehydrated, right, that you go into shock. So replacing electrolytes and fluids is the best treatment and aid other than treatment of the actual bacteria itself. So water purification, hand washing, all of these things really help in our prevention. They even supposedly have a vaccine for this, although I've never heard of anyone getting the vaccine. And what else did I want to say about this one? Right, obviously we're ingesting contaminated food or water. Remember, we talk about most of the diarrheal diseases, it's what we call the fecal-oral route, where unfortunately fecal matter has gotten into your water or your food and you've ingested this organism that way. That's how it's transmitted. Sometimes in natural sources associated with marine crustaceans. So the major problem here in Louisiana is shellfish, particularly filter feeders like oysters, right? And that's why we have all the specific regulations we have for testing the waters, how the oysters are transported. That's why a lot of warnings in restaurants, if you eat it raw, right, you are at risk. But we do the best we can to minimize that risk. And so in 2006, you'll notice I left this graph up even though it's kind of old, but because they talk about that particular instance as it applies to seafood. Where if you look at the current numbers, last year we had 0 cases of cholera. And most of the cases come from people traveling to other parts of the world. And I think we had mentioned last time, I had looked up that Haiti was where most of the people were getting it, and I couldn't remember if that was when the tsunami was in 2011. That I didn't look up, but you could easily look up, and it would make sense if that much higher incidence would be related to an event like that, and a lot of relief workers heading to that area and then coming back to the United States. For shigellosis, this one tends to be much more serious. It can lead to dysentery where you have the bloody stool. Again, because it has the ability to kill our cells with the toxins that it produces, it also enters into our cells. This is a really nasty bacteria. And there are actually several species of bacteria that can cause this infection. Dysentery, that species is one of the worst. So in addition to gaining entry into our cells, one of the unique things that this one can do as well is that notice our M cells normally are going to transmit these guys to the macrophages, but they can escape out of the macrophages, and they can even, once they gain entry into our cells, they can travel through our cells, and it almost looks like in these drawings, right, that they have flagella. No, what they do is they can manipulate and activate our actin filaments inside of our cells, part of our cytoskeleton, and they basically use this as a slingshot to propel them from one cell to another, right? So in this picture, that's what they're showing you here is the actin filaments that they're attaching to and activating to be able to travel through our cells. And then, of course, as our cells die, as our cells are deteriorating, we have neutrophils, we have inflammation in that area, and quite a bit of destruction. So the toxin associated can cause really fatal problems when it gets to your kidneys as well. It can cause the kidneys to shut down, so you can end up with what's called hemolytic uremic syndrome. So this can be quite serious infection. E. coli, some different pathogenic strains of E. coli can also be damaging. What's wrong? Oh, okay. Oh, yeah. So again, right, very similar set of circumstances. And for this one, it really depends on the pathogenic strain that you ingest, what toxins they have at their disposal. And so Shiga toxin is a toxin produced by Shigella. I'm trying to see if I have it on here. No. So we have the different degrees of differences between the different E. coli's, the different pathogenic strains. So enterotoxogenic is a term used for ones that produce two kinds of toxin, one that's similar to the cholera toxin, right? And so therefore very similar symptoms to when you have cholera. Enteroinvasive, these guys tend to enter into and can cause destruction of the epithelial cells of the intestines. Enteropathogenic, these ones the attachment of the bacteria is followed by the loss. So these are the ones that are able to create that pedestal for them. And then the worst case is the Shiga toxin producing. These used to be referred to as enterohemorrhagic, right? But recently it's been changed to Shiga toxin, so it's abbreviated STEC for the MWR report. And the most common serotype that causes this this really serious form of gastroenteritis caused by, you could even call it dysentery, that produces this particular toxin is E. coli 0157. Serotype meaning that they are using antibodies to detect the particular sugars associated with that outer membrane. That's the O part, right? And this one is the particular number sequence in this typing that they do. So O157H7 is the most common offender that produces this. And so we have substantial outbreaks of this in the United States. So this is the 2006 distribution. You can see there's no direct pattern, right? It's kind of scattered throughout the United States at any one time or another. Although this particular one for 2012, it really seems like the larger problem is in the upper states, but it really isn't a regional thing. It just depends on where the outbreaks are happening, what's been contaminated with it. So some outbreaks that have happened in the past are like spinach. Remember when they pulled spinach from the shelves? And it was because a local dairy farm nearby had runoff that was running off onto the fields, and that's how the spinach got contaminated. So there's been several. You keep you really busy with all the different E. coli outbreaks, but we'll move on to Salmonella. And the interesting thing with this one is that it's very common with poultry, right, that they have this bacteria naturally, okay? So sometimes I'll have increased numbers, and this is why you always want to cook your chicken, right, poultry products all the way. You don't want to eat raw eggs unless they've been pasteurized as well. And so because it can contaminate other types of food as well, and they look at shigellosis along with this one again, because a lot of times it's foods that are contaminated. So this particular spike they were able to attribute to a particular outbreak of pasteurized milk that was actually contaminated with salmonella. But as you'll notice, numerous things, right? There in 2006, there were tomatoes that were contaminated, fruit salad, pet turtles. Turtles always have salmonella, y'all, right? For the most part, it is something that they carry. So always, always, if you have pet turtles or deal with turtles, always wash your hands. Always, always. They actually have state regulations in the state of Louisiana on what size a turtle can be when it's sold. And supposedly they have to be big enough that the kids can't put them in their mouths. But if you're a parent and you're letting a little kid put a turtle in your mouth, you've got issues, right? Okay, but yeah, I guess to help with, preventing infection. So as you can see, it's been pretty steady incidence-wise. Like we haven't had any significant, huge outbreaks, but we've had numerous outbreaks of both salmonella and shigellosis. Although like I said, according to this graph, we're doing pretty good stable-wise. But again, notice small turtles, live poultry, even hedgehogs. I hadn't seen them on the list before. So Campylobacter jejuni, right, another bacterial infection. Large foodborne and waterborne outbreaks are usually originating from chickens, cows, and other animals, although person-to-person spread rarely occurs. Can't say enough, right? Cook your food, keep your work area clean. So that's it for bacteria. That brings us to viruses. So for prevention of hepatitis A, B, and C, which ones do we actually have vaccines against? Anyone know? Which one can you not get vaccinated against? C. There are vaccines for both hepatitis A and B. So how do we know if you've had these infections? One of the ways sometimes that we could test is to look for the antigen or the antibodies in the blood of a hepatitis victim. And so when they test blood, right, at the blood center, when you donate blood, they test for these things. And if they find anything in your blood, they will alert you as well, right? So I've had friends and relatives donate blood, and then they're like, okay, you can't donate blood anymore. You've had hepatitis. We don't want it. But I don't know how that goes with the vaccinations that they're doing now. So as I said, although A, B, and C are the main ones that we talk about, and these are viruses, the difference here is that notice that hepatitis A and C are single-stranded RNA viruses. Hepatitis B is actually a double-stranded DNA virus. We do have vaccines, as we said, for both hepatitis A and hepatitis B, as well as we can give immunoglobulin treatment, right? We can give someone antibodies to help treat. Hepatitis A, a lot of people in the past, before we had vaccination for it, would get hepatitis A. And the bad news with that is that you can be sick for quite a long time. It takes quite a long time for your body to recuperate from this type of infection. Hepatitis B, on the other hand, much more serious, can cause progressive deterioration of the liver. Hepatitis C is one of the more relatively new ones, and the sad news with this is we don't have any vaccination for it yet. Don't know if we will be able to achieve that. Where hepatitis A is fecal-oral route, so this is something that could contaminate your food or water. You're usually only going to come in contact with B or C. If you come in contact with blood, possibly semen for hepatitis C, but for hepatitis B as well. Right, so your exposure should hopefully be limited. Hepatitis C, again, was transmitted at one time by blood transfusions because we didn't know it existed, right? We couldn't test for it, right? And now we have the ability to test for it. And since then, you know, incidence has significantly decreased. Hepatitis D, another single-stranded RNA virus. Again, blood or serum. This one is not very common. You don't hear very much about it. Oh, wait a minute. This isn't updated, is it? It must be D. Yes, it is D. Okay, so the causative agent is wrong here. It's actually not a virus. It's a viceroid. It's a piece of single-stranded RNA by itself, and it typically infects the hepatitis B virus, and that's why nowadays, if you have hepatitis B vaccination, it helps protect you against D, because hepatitis D is actually inside sometimes the hepatitis B virus, right? So there's a virus. So that's relatively new information on hepatitis D, that it is not actually a virus. It's a viceroid. I won't ask you guys that on the test or anything. It's new stuff. But I just noticed that. I was like, I need to find a more up-to-date table. I don't know if obviously they didn't update it in that version of the textbook. Hepatitis E and G, again, not very common in the United States, these particular types of hepatitis. Notice E is also fecal-oral like A is. And again, G is blood, possibly semen transmitted.
I have a question about Hep C.
Mm-hmm.
When my husband was a teenager, he tested positive for it,
Mm-hmm.
and years passed, and now it's just not there. Could it be dormant, or maybe it was a false positive, or?
Could be any number of things.
Because he's never had like any liver problems.
Right. And I'm sure they hopefully would have continued to monitor him.
Yeah, he gets
Yeah.
blood tests all the time.
Yeah.
He goes for his job, and nothing ever comes up.
It might have been a false positive considering it was one test.
I hope so.
Yeah, I hope so too, because it can be quite debilitating to the liver. So as you can see, data-wise, right, hepatitis A vaccine was licensed in 1995. Since then, in the use of the vaccine, the numbers have significantly decreased, right? Used to be, like I said, at one time, just about everybody ended up with hepatitis A at one time or another. Hepatitis B vaccine was licensed in 1982. And again, notice the line doesn't really start here for hepatitis C, right? Because the longest time we didn't know about its existence. So it wasn't until we get a test for it and start collecting data for it. But the good news is that because it's not transmitted very easily, right? You had to come in contact with blood, possibly semen. The knowledge of that and the blood tests that we do, we've seen a distinctive decline in the number of infections and not a rise, which is good. The last is protozoal diseases of the lower intestinal tract. So these guys are all caused by protozoa, right? So giardiasis is caused by a protozoa, cryptosporidiosis, cyclosporiasis, and amebiasis. So what do we know, what's similar about these guys' epidemiology for these protozoal intestinal diseases, and what's different, right? So most of them, right, their causative agent is kind of in there, is their genus name for the most part, except for amebias. Entamoeba histolytica is the most common out of the entamoeba. How would we prevent these types of infections is what we want to really think about as we look at these. So, as I said, they're all protozoa. Giardia labia or instantalis, it has been, I think, officially changed to instantalis instead of labia. It's a pear-shaped protozoa that has multiple flagella, has two paired nuclei, so it almost even looks like it has eyes. It doesn't. Typically what you do is you ingest the cysts, which can survive passage through the stomach, right? So the stomach acids don't kill it. The trophozoites emerge from the cyst in the small intestines where they attach. They have like these little sucking discs and they attach and they suck your mucus. Mucosal function is impaired by the adherents as well as the protozoa and the immune response. Ingestion of fecal contaminated water, person-to-person transmission, particularly in daycare centers, can happen. So how do we prevent? We boil or disinfect drinking water. We filter, right, community drinking water supplies. And then for treatment, right, there are different types of drugs that are used to treat. So, not actually that interesting of a picture for these guys, but there's them swimming through. So, cryptosporidiosis, another protozoa. Again, notice following ingestion of oocytes, right? So this is a protective form. And then when they get to our intestines, our intestinal juices actually digest away and they release the 4 banana-shaped sporozoites, which then invade our epithelial cells, causing deformity and to your intestinal alveoli. And then we get inflammation in the area, we get secretion of water and electrolytes increase. Our cell-mediated immunity usually is able to take control of this, because they actually get inside of our cells, right? So cell-mediated response. The oocytes are infectious when discharged within the feces. The infectious dose is small, right? Because again, they're kind of protected in that oocyte. So when you ingest them, they go right through the stomach and they're not affected. Person-to-person spread readily occurs in unsanitary conditions. Infected individuals can discharge the oocytes for weeks after symptoms subside. The oocytes are resistant to chlorination, and they pass through many municipal water filtration systems. The host range is wide, including cattle, pigs, and dogs. So this is one of the reasons why pasteurizations of beverages such as milk, apple juices, bottled drinking water, filtration has to be with a really small filter, right? Really small pore size. Sanitary disposal of human and animal feces really helps, right, with keeping stuff from being contaminated. Cyclosporidiosis, so again, another protozoa, another one with oocytes that are not infectious when discharged in the feces, though. And so person-to-person spread does not occur. Travelers to tropical countries are at risk for infection. So produce, especially raspberries imported from tropical Central America, has been implicated most in North America for the outbreaks. So this is not something where it can really get in, right, and we can't wash it out. Use of bottled or filtered drinking water is advised in the tropics. Thorough washing of imported berries, leafy vegetables. Most victims are affected most victims are effectively treated, right, with drugs. Entamoeba histolytica, as I said, is the most common causer of amebiasis. Again, this one's a cyst former, so the cysts are liberated in the zoocytes in the small intestine upon reaching the long intestine. The trophocytes feed on the mucus and cells lining the intestines. So remember, these are amoeboid, so they don't have a particular shape to them, right? They can move by pseudopods. They just basically stick to your intestines. The enzymes produced that allow the penetration into the intestinal epithelium and sometimes the intestinal wall and blood vessels. And tendency to the liver and other organs results in abscess, so it can actually travel through the body. Ingestion of fecally contaminated food or water, diseases associated with poverty, homosexual males, and migrant workers. Good sanitation and personal hygiene is one of the best preventative measures. I'm not sure which amoeba they had in the water supply. What was it, in Jefferson Parish here, where they had a problem with it for a while? Yeah, I think it was a different one. So what's the take-home mention with prevention? Avoid contaminated waters, right? Definitely water, right, or food that's been contaminated by water, right? Different ones, different areas, right, that you have to worry more so about them. But definitely want to avoid. Let's see. Yeah, we need to at least start the next one.